proceed with the rest of the lecture on plant hormone. So we're going to start at 9:10. We'll wait for maybe some of you to join in, right? Right. We shall start now. So yesterday we have already talked about auxin, plant hormones, what are plant hormones, and basically the five classes of plant hormone itself. And we have talked extensively about about auxin. We not just look at the role of auxin in elongation of the shoot tip and also the growth of the roots, but we also look at how diverse the the role of auxin in plants. So that's why auxin is one of the most researched hormone in plants. So today we are going to proceed with the other hormones, right? Share my screen first. So we're going to proceed with another hormone. So we're going to talk first on so we are going to talk about gibberelline or gibberellic acid and what is it about in terms of why is it important and how can we manipulate the gibberellic acid that we have in algae culture. And gibberellic acid, I'm not really sure whether you guys have heard about it or not, but um,
Recording in progress.
always is associated with um, germination state, right? So we are going to look at gibberellic acid first. Gibberellic acid or gibberellin are basically made up of a lipid structure. It's a derivative of lipid. It specifically it's um, synthesized from diterpene, right? From terpene biosynthesized from geranyl diphosphate, a common C20 precursor for diterpenoids, and it diverse aspects of growth and development, which include pollination, and it also works together with auxin, controlling stem elongation, flowering, fruit development, and the regulation of gene expression in the cereal allurin layer. And basically, when we talk about gibberellic acid, it can also be, you know, used in the industry, in the brewing process, right? This is the structure, how it looks like. It's a gibberellic acid, right? You can see because why is it a gibberellic acid? Because it's made up of fatty acid derivative, which is diterpenoid itself, right? So gibberellic acid occurs flowering, we call it as angiosperm plants, and non-flowering gymnosperm plants, fern, mosses, algae, and even fungi. So there are over 100 analogs identified, can be identified from plants, fungi, or bacteria. So they are used commercially in the agriculture industry to initiate the seed germination by breaking down dormancy, uh, which eventually leads to the growth of the seed itself, and it promotes fruit setting in a number of plants, including the production of partenocarpic or seedless varieties and to produce dwarf plants. So it works together with auxin for partenocarpy. And it can also be used in the beer industry. We're going to look into that later on, right? So what are the effects of gibberellin? Like I told you guys just now, uh, it also plays a role in controlling the growth of plants. Um, if auxin also control growth of the plant itself, but for gibberellin, it controls immature plants, right? It is produced by roots and young leaf. It increases growth by both division and elongation. It promotes elongation of the dwarf mutants. It stimulates shoot elongation even in mature regions of tree and shrubs. And it also basically has effects on seed germination. So when we talk about gibberellic acid, it's more about seed germination itself. Breaks dormancy of certain seeds. Because of this function, it is then manipulated in the beer industry, using it to induce the growth of the seed. Right, the seed would break from dormancy and would release a surge of carbohydrates or monosaccharides that can be used by single cell organism like yeast and feed on that and eventually would produce alcohol, right? That's how it is used in the industry. And this is an experiment done in order to show how gibberellic acid also can affect growth in dwarf plants. All right, so we have control here without any treatment, and we have control. We have dwarf plant with treatment of exogenous or external source of gibberellic acid. You can see that the difference. You can see that there is more growth uh, in the dwarf plant that has been exposed to gibberellic acid. Right. So gibberellic acid works on dwarf plants. So it induces the growth of the dwarf plant. That's why it can also be used to sort of add more value to a dwarf plant, making it growing more by applying exogenous gibberellic acid. And then we can see here there are also experiments on another plant, not a dwarf plant, right? So we can have a normal plant here um, without any treatment. You can see the growth is just like this, all right? And wild type corn is tall and its height is unchanged by application of GA. So this one here is what we call as a corn plant. right? A normal corn plant would just grow like this. Tall plant is not a dwarf plant. And then when an experiment was done, uh, supplying more and more gibberellic acid from the external source, you can see that there is no effect, right? When you compare it without any treatment. So, like I told you guys yesterday, hormones have their own receptor, right? Just like how enzyme has their own active compound. So they have like a certain capacity to take up the hormone itself. So only a certain amount or certain concentration of hormone can bind to the receptor. And this is the case with normal corn. Apparently, normal corn produces enough gibberellic acid to saturate its GA receptor. So when you supply an exogenous gibberellic acid, it won't cause any effect, any significant effect, because the receptor is already full or saturated with the original gibberellic acid, right? So the exogenous gibberellic acid cannot bind to the receptor anymore and cause any changes. So the experiment itself, in conclusion, we can say that um, gibberellic acid also supports growth and development of a plant. So gibberellic acid can be used in beer industry. Uh, there's no there is no way I'm supporting beer industry, but it's just for knowledge wise, all right? So GA is used in beer industry in the terms of it basically breaks dormancy, and it is used in grains such as in barley, all right, um, in the aileron layer. So gibberellin from the embryo of germinating grains are necessary for the synthesis of alpha amylase by the cell of the aileron layer, which in turn is necessary for the hydrolysis of starch within the endosperm. So gibberellin is produced by the seed itself, and it will then enters the aileron layer. And this aileron layer, it has starch, storage of starch, proteins, which can be used um, to release monosaccharides, right? And the hydrolysis of starch basically requires an enzyme known as alpha amylase. So gibberellin acid or gibberellic acid basically enters the aileron layer, enters the endosperm, and then after that diffuse itself into the aileron layer. And this aileron layer contains many starches or starch granules, right? That contains uh, carbohydrates, okay, the store polysaccharide storage. And then in order for the seed to break dormancy, it needs to hydrolyze the starch, supply energy, and it needs the enzyme of amylase. So aileron layer protein stored as granules in the cell of plant seed regulation of gene expression. Material aileron layer. Later on, we're going to look into that. So in the brewing industry, the production of beer relies on the hydrolytic breakdown of starch and barley grains to yield fermentable sugars, principally maltose, which are then subjected to fermentation by yeast. So during fermentation, glycolytic enzyme from yeast break down the sugar, resulting in ethanol. So basically, I'm just going to use the figure here to explain more to you about how uh, gibberellic acid plays a role in barley seed germination, which is then manipulated for the beer industry. So this is a barley seed, right? We have endosperm here, a starchy compound and a starchy structure, right? And we can see here, this is how it looks like. This is a coleoptile, and we have shoot apical meristem, first foliage leaf, and when it's the time to break dormancy, it's basically the seed would absorb water, so gibberellic acid production will then be increased, and this gibberellic acid will then diffuse into the endosperm, alright? And then after that, it would then go to the aileron layer here, and this aileron layer contains aileron cells that contains starch as a carbohydrate storage, and this aileron cell will then produce alpha amylase, and this alpha amylase will then be used to hydrolyze starch and other macromolecule, which in turn would produce a monosaccharide or a disaccharide, such as maltose. All right, maltose is a disaccharide made up from glucose components or glucose molecules, and from that they sort of put yeast, and this yeast here can definitely feed on that maltose and undergo what we call as alcoholic fermentation, producing alcohol in the form of ethanol. So if you can still remember your carbohydrate metabolism, we know that in carbohydrate metabolism we have glycolysis. Glycolysis would then produce pyruvate, and pyruvate would either go to the Krebs cycle. If there is not enough oxygen, it would opt for lactic acid fermentation. But one single cell named as yeast, it can also undergoes what we call as alcoholic fermentation. Why is it called alcoholic fermentation? Because eventually it would produce ethanol as the final product. And this is what we call as alcoholic fermentation. And this process is always used and manipulated in um, beer industry or alcohol industry, right? To mass produce alcohol, right? So this is just as a refresher of what alcoholic fermentation looks like and what are the enzymes involved. And that's why I put the slides here, just to try and refresh back your memory on alcoholic fermentation and what are the enzymes involved. And these enzymes, you can find it in the yeast itself, right? So gibberellic acid basically it gives signals to the endosperm to break uh, from dormancy. And in order to break from dormancy, it needs energy. So it needs to hydrolyze the starch storage that it has in order to release sugar. And in the beer industry, the sugar that is being produced by the barley seed here is then um, sourced to yeast. And then yeast will use this and undergo alcoholic fermentation. So that's how um, the role of gibberellic acid is being modulated in the um, beer industry. So in the industry itself, they can also supply exogenous gibberellic acid. So this exogenous gibberellic acid will then increase the rate of uh, germination. So the regulation of metabolite mobilization by gibberellic acid is a complex process that involves not only the synthesis and organization of protein export machinery, but also the initiation of the expression of a number of genes encoding hydrolytic enzyme. So just now, we look at gibberellic acid being produced by the coleoptile itself, and eventually it will go to the starchy part of the seed. And how does it induce germination is by inducing or stimulating the expression of enzyme that is involved in hydrolysis of starch. And in this case, we are talking about alpha amylase enzyme. So alpha amylase enzyme are produced by the iron cells in response to the hormone, which is gibberellic acid. And these alpha amylases have been the most extensively investigated of the hydrolysis produced by germinating barley seeds. So gibberellic acid does not only induce expression of one gene of alpha amylase, but many types of alpha amylases. So, we know that there are basically four alpha amylases, and these four alpha amylases are made up, as you guys know, enzymes are made up of proteins, and proteins are made up of amino acids. And because of this amino acid composition, these alpha amylase is being distinguished from each other by their isoelectric points. And seed germination, it, it takes time, all right? As you can see here, there are four alpha amylases that have different isoelectric point, and because they have different isoelectric point, they are being released according to time. So the too high isoelectric point or the too high alpha amylase, right? reach their maximum abundance approximately 12 hours after gibberellic acid treatment, whereas the other two alpha amylases with comparatively low PI value show a slower development and do not reach their maximum expression until 24 to 32 hours after gibberellic acid stimulation. So what they are saying here in the statement, there are many alpha amylases that can be induced by gibberellic acid, not just one. Okay, so when external or exogenous gibberellic acid is being introduced to the barley seed, it would induce the expression of gene involved in um, forming four types of alpha amylases. And these four types of alpha amylases, they have their difference in terms of isoelectric point. So what is an isoelectric point? So I believe you guys have learned this in your biomolecules, in proteins. So you have to find out what isoelectric. And this isoelectric point is the one that determines which one of the alpha amylase would be released first when it is exposed, when the seed is exposed to exogenous gibberellic acid. So the first two amylases with high PI value, it basically would um, be induced first in the first 12 hours. And after that, the one with low PI value will then have like a sustained release, right? Up until 24 to 72 hours. So by doing this, we know that there is a continuous supply of alpha amylase that can basically induce continuous hydrolysis of starch in order for the seed to germinate and produce maltose. So alpha amylase activity continues to accumulate in the medium for a long period when allured cells or protoplasts are treated with gibberellic acid. So this is how gibberellic acid is used in the industry. And if we move on to the next slide itself, there's also like an experiment being done looking at the barley seed germination if there is exogenous gibberellic acid being supplied to the seed itself. So we have three parts here, or three treatments. So always in experiment, you have to have control in order to do comparison, in order to know that you are doing things right in an experiment. So you can see here control, the alpha amylase production is at normal condition, no sudden increase or no sudden surge, all right? And then when there is a supply of exogenous gibberellic acid at just 1 micromolar, you can see that there is an increased production of alpha amylase. And it's like a significant increase from 10, maybe 5 micromolar here, or uh, five-fold, not five-fold, 5 units, all right, of alpha amylase in the control. And then when we supply with 1 micromolar gibberellic acid, it basically increase up to ninefold, right? 9 or 10-fold, like that. And they also did an experiment where they supply 1 micromolar gibberellic acid, and also they supply an inhibiting hormone, which is known as ABA, or abscisic acid. So abscisic acid is all about abscission, it's all about inhibiting growth. So that's why you can see that they, the abscisic acid basically suppress increased production of gibberellic acid. So the appearance of the alpha amylase enzyme can largely be blocked by another hormone called abscisic acid. So this is just to show you guys that how what is the role of gibberellic acid in barley seed germination. It basically increase the secretion of an enzyme known as alpha amylase. And how does it increase? This is by inducing more gene expression, gene that basically is responsible for alpha amylase activity. Right. So we're going to look further in terms of um, what are the things that gibberellic acid basically induce. how does it induce the expression of the gene itself. Okay. So yeast needs a large supply of malt sugar or maltose from barley to ferment and do alcoholic fermentation, producing ethanol. So as the seed absorbs water, the embryo produces gibberellic acid. Gibberellic acid then attached to a protein receptor, GID1. It is a G protein receptor, and it triggers the destruction of what we call as DELA protein by proteasome, and gene transcription begins to switch on GA-MIP genes. So this is the gene that are responsible for transcription of alpha amylase mRNA. So through transcription and translation, this leads to synthesis of amylase, which then is excreted into the endosperm, stored food reserve. Amylase breaks down starch to glucose unit, which diffuses to the embryo and are used for plant growth. And this lengthy description is explained throughout the figure here. So this one here, gibberellic acid, and then it binds to a receptor here. You can see there's an interaction between the hormone itself and also the site of the receptor. And then this receptor is what we know as this one here, GID1, and it's basically it's coupled with GTP, right? When it's coupled with GTP, it will then um, induce production of a signal, and this signal will then be transported inside the nucleus so that it would affect the gene expression. And in inactive form, all right, we have a DELA protein here. So DELA protein would bind to the gene that basically encodes for amylase, which is MIP-B or MYB, right? Or MyB. Okay. So when gibberellic acid binds the receptor, it transports signal into the nucleus, and this signal will then bind to the DELA protein and release the DELA protein from the gene, making it active, right? Making the gene active. And then after that, it would induce transcription of the MIP gene, right? Making more mRNA, hence will be translated into protein. So you can see the mRNA for alpha amylase, which eventually would be converted into protein, right? Alpha amylase is then uh, transported out from the nucleus by rough endoplasmic reticulum and Golgi apparatus here in a vesicle form, and then it would release to the endosperm, which is rich in starch. And in endosperm, it will then hydrolyze the starch. So this process here basically happens in the alluron layer by conducted by the alluron cells. And these alluron cells release that alpha amylase and then would um, release it into the endosperm. So we go back to the previous figure here, all right? So you can see gibberellic acid, it's synthesized by the embryo, and then after that, it the alluron cells in the alluron layer to do what we call as gene expression, increased gene expression of MIP, right? To encode for more alpha amylase enzyme. And the alpha amylase enzyme produced by alluron layer would then be transported back into the endosperm, all right? And this endosperm contains a lot of starchy components, and the alpha amylase would then hydrolyze the starchy components into maltose, and this maltose can then be uh, channeled to the yeast, right? To yeast, and yeast can use that as a source of energy and do alcoholic fermentation to produce ethanol. And that's how uh, gibberellic acid is being used in beer industry, right? Other effects of gibberellin. So just now we talk about how gibberellin has effects such as extensive growth and seed dormancy, right? The other effects of gibberellic acid we know as juvenility and flowering. So juvenile stages of some plants have different shapes of leaf than the adult stage. So gibberellic acid have determine whether a particular part of a plant is juvenile or adult. For example, the buds of adult branches grow into adult branches, but if treated with gibberellin, it grows into juvenile branches. So the plants would keep on being juvenile, right? If gibberellic acid is being uh, supplied to them, it's just like the juvenile hormone in insects, where juvenile hormone is being produced in order to keep the insects uh, from being an adult insect at the wrong time. So it's the same thing with gibberellic acid, the function, the effect here. And then flowering, gibberellic acid induces flowering. So you can see that if a plant can uh, can produce flower biennially, right? When we supply gibberellin acid, it can induce flowering. So the plants can have like a flowering every year instead of every 2 years, which is biennial, right? And then the fourth or the fifth one is partenocarpy, and the effects of gibberellin here, it works together with auxin. It stimulates pollen germination and growth. It results in larger fruits and even bigger flower and fruit formation. It increases size of fruit. So because gibberellic acid it induces growth, so that's why when you apply or when you supply gibberellic acid, it can cause production of larger fruit. It can cause production of um, yeah, larger fruits and also bigger flowers. So this one here um, is a vineyard, right, or a grape orchard. You can see that this is the normal size of a grape. Okay, and then this is after gibberellic acid treatment. You can see that it produce more fruits, and the fruits have are bigger in size, right? And it works together with auxin to induce what we call as parthenocarpy, which is production of fruits without fertilization. So no seed in this fruit here. It can either be in grapes, it can either be in cucumber, tomatoes, so on and so forth. Right? So if in Malaysia we have seedless guava, all right? So gibberellic acid and auxin work together to induce what we call as parthenocarpy. So it's a process tightly controlled by plant hormone, and parthenocarpic fruits can be obtained by the combined exogenous application of three enzymes here. We have auxin, we have gibberellic acid, and cytokinin. So cytokinin is the one that induce division of the cell itself. Um, hormones, auxin, gibberellic acid, and cytokinins, especially in the first two, are well known to induce parthenocarpy. There's auxin treatment of young unpollinated ovaries and certain cultivars of strawberries, tomato, grapes, and orange is known to cause production of parthenocarpic fruits. Also, treatment of certain cultivars of oranges, tomatoes, blueberry, garden pea, and Arabidopsis by gibberellic acid 3 or gibberellic acid 1 causes parthenocarpic fruit development. Okay, so this GA here, it supports the growth of the fruits. So auxin induces parthenocarpy, and the fruits produced will then grow in its normal size or grow bigger by application of gibberellic acid. So these hormone treatments are given to ovaries of unopened, unpollinated, or emasculated flowers just before anthesis, during the receptive periods of anthesis. So parthenocarpy is basically the ability to develop fruits without pollination, without fertilization of the ovule, and therefore without seeds development. So anthesis in this case is the flowering period of a plant from the opening of the flower bud. So it's a period where plants start to flower. So it's called anthesis. So you can see here, this one here, it's a research article being published in 2007, looking at the role of gibberellic acid and auxin on supporting parthenocarpic fruit growth induction. All right. and how they work synergistically with each other to support the uh, parthenocarpic growth induction. So the effect of applied gibberellin and auxin on fruit set and growth has been investigated in tomato, CV microtome. It was found that to prevent competition between developing fruits, only one fruit per trial should be left on the plant. So they have a plant, a tomato plant, and then they just leave one fruits only for each plant, and they would observe, right, the effects when applying uh, exogenous gibberellic acid and oxide. So unpollinated ovaries responded to GA3 and to different auxin. So they applied the indotriacetic acid, naphthalene acetic acid. These are all resemblance of auxin. And then we have the synthetic auxin that we have talked yesterday, 2,4-D. So 2,4-D, like what I mentioned yesterday, the structure is more stable. So it basically persists more or absorbs more by the plant itself. Okay. 2,4-D being the most efficient induced fruit had different internal morphology with poor locular tissue development in the case of GA and pseudoembryos development in the case of 2,4-D. So one hormone basically induce parthenocarpy. Another one supported by increase or induced growth in fruits or flowers. Okay, so parthenocarpy is without pollination. So what are the difference between auxin and gibberellic acid? So I told you guys just now, in terms of parthenocarpy, it works together with auxin. But gibberellic acid also supports growth. When it supports growth, it's basically also involved in elongation, differentiation, and division of the cell itself. So gibberellin control or gibberellic acid controls elongation in mature region of trees and shrubs, whereas auxin regulates elongation in grass seedlings and herbs. So auxin is on grass seedlings, but gibberellic acid can also control growth in mature region, right? In mature regions of the plant itself. So plant can tolerate a high level of gibberellin, but not of auxin. So remember, uh, auxin is only needed at low concentration in shoot tip and also in the growth of roots. So if basically auxin is in high concentration, it would the the effect be reversed, right? Instead of supporting growth, it will not support growth. It has high concentration of auxin. Gibberellin, on the other hand, has little effect on root, whereas auxin has more of an effect on roots itself. So these are the differences between auxin and gibberellin. So one thing that you guys have to know, gibberellin also support growth, all right? But what are the difference with gibberellin and auxin? These are the different. One is inducing mature region of the trees and shrub itself, and gibberellin can be tolerated high concentration, and gibberellin has less effects on roots compared to auxin itself. Right? So that is gibberellic acid. Another one that also support cell division, support cell growth is what we call as cytokinins, and this is how it looks like. Um, it promotes cell division. It resembles um, adenine. So this is one of the cytokinins that has been identified in plants. It was first identified in Z maize or in corn. That's why it is called zetin here. And it has a distinct structure, ring structure here, just like adenine. And it also promotes cell division and have other similar functions to kinetin. So naturally, there are 200 naturally and synthetic cytokinin. 200 of them have been identified. The most common form of cytokinin in plant is called zetin, which was first isolated from corns or Z maize. Concentrations are highest in meristemic region. So meristemic region is the region in plants that undergo rapid cell proliferation, rapid cell division, right? Within roots, young leaf, developing fruits and also seeds. So it supports cell division. Hence, it supports the growth and development. And it works together with auxin and with gibberellic acid in what we call as parthenocarpy. And it can also work together with auxin for cellular division. So effects of cytokinins, it comes from the word uh, effects of cytokinins, basically division. It controls cellular division. So cytokinins stimulate cellular division by speeding the transition of cells from the G2 phase to the M phase of the cell cycle. Just like human, just like animal, plants also have their own cell cycle. It's the same phase. All right, we have G phase, we have M phase, and then S phase. So it's the same in plants. And the role of cytokinin itself, it speeds up from the growth phase into the mitotic phase, which involve lots and lots of division of the cell itself. And all this is dependent on this presence of auxin. So we have, if say for example, we have auxin alone, cell will only expand. And then if we have kinetin alone or cytokinin alone, it has little effects. But when we have both auxin and also cytokinin, it would induce synergistic effect. It would support rapid cell division and cell proliferation. So effects on cotyledon, it promotes cellular division and expansion in cotyledon, increase in wall elasticity from cytokinin, result in cellular expansion. And because of that, because auxin, if you guys can still remember yesterday's lecture, how we see that auxin induce elasticity of the cell wall, hence induce what we call as uh, expansion of the cell, elongation of the cell itself. So that's why cytokinin also work together with auxin in supporting cellular division, expansion, right, in cotyledons. Uh, so this is just to show you the cell cycle in plants. And we start off with G1 here. G note if something happens to the cell itself. So try to remember your growth factors lecture, how I showed you guys the cell cycle and what does it mean by G note here, right? Then we can see that cytokinin works to accelerate the transfer from G2 into mitosis or mitotic phase, the M phase. So it would basically increase the rate of cellular division and cellular proliferation. Besides that, cytokinin is also responsible for formation of organs in plants in a process known as organogenesis. So basically means organ generation. So cytokinin again it works together with auxin to affect organogenesis. So high cytokinins auxin ratio favors the formation of the shoot itself, right? And low cytokinin auxin ratios favor the formation of roots. And cytokinins also has effects on senescence. It delays the senescence. Cytokinins delay, because cytokinin is all about cell division. So in the case of senescence, it delays the process of senescence, just like how auxin delays the process of senescence itself. So cytokinins delay chlorophyll breakdown in the dash leaf by preventing those genes from being turned off that stimulate the formation of chlorophyll. Okay, so we have a detached leaf or abscissed leaf. Okay, so cytokinin would delay the chlorophyll breakdown if it is applied to the detached leaf. And when it is applied to the detached leaf, it would turn off, um, it will prevent the genes that are involved in the formation of chlorophyll from being turned off. So that's a continuous maintenance of the chlorophyll in detached leaf. It also promotes the conversion of AQ plus into chloroplast via stimulation of chlorophyll synthesis. So that one is for cytokinin. So it basically works together with auxin. Whether I have missed anything or not. So now we go to ethylene. All right, ethylene is basically, if you still remember your biological chemistry, ethylene is a standard or common name for this compound here, which is ethene. All right, it is called ethylene, and it occurs in the form of gas, and it's basically a result of uh, degradation of methionine here, and it would release ethylene. So in 1901, Dmitry Neljubov, a Russian scientist, recognized ethylene as a plant regulator. Later on, we're going to look at the experiment that this scientist has done. In 1934, Arjun fully identified ethylene as the first cases plant produced hormone. It's found in tissues of ripening fruits, nodes of stems, senescent leaf, and flower. So ethylene is mostly known to induce ripening of the fruit itself. So this is how the structure looks like. Very simple structure, and it occurs in the gas form, right? So the first experiment to identify ethylene was done by Neljubo. So Neljubo appears to have come to his revelation about ethylene through the careful study of germinating pea plants inside his lab, right? So this one here. So basically what Neljubo did, he put a pea, right, seed in a black box. Okay, and it has an opening here. So one is exposed to the air in the lab. Another one is exposed to air outside. He put it just next to the window so that the opening would only allow entrance of air from the outside. And he compared it, all right? He compared it between the rate of germination of the seed itself. Okay, so what he found out that the peas exposed to the air in the lab would uh, would swell up, right? So he implanted peas in a pair of pitch black boxes. Into one, he pumped air from the outside. The other one, he fed air from his laboratory. So those pea fed the laboratory air grew sideways and swell up. It grows, it ripens up. He then isolated ethylene found in the illuminating gases burned by lamp in his lab and on the streets at night. So eventually, he identified that the ethylene, or there is like a compound or a gas being produced from the illuminating gases burns by lamps. So this experiment happened in 1901. So they use gas to burn the lamps instead of electrics. All right. So from the gas is where it finds ethylene, and this ethylene is the one that causes the uh, growth of the or germinating of the pea plants, and that's how Neljubo basically says that it induced what we call as ripening or swelling of the pea by ethylene. So when we talk about ethylene, basically associated with fruit ripening. So in fruit ripening, ethylene stimulates all these factors of fruit ripening, breakdown of chlorophyll, and it would synthesize other pigments, other accessory pigment such as lycopene in tomatoes. We have carotenoids. Instead of doing chlorophyll, the plant would now focus more on synthesis of accessory pigments that support fruit ripening. It would produce more colors in the fruits. That's what happens. and then fruit softening by uh, cellulase and pectinase uh, formation. So basically, when we have ethylene, it induces fruit ripening. And how it induces fruit ripening is through these processes here. So the first one is breakdown of chlorophyll. So chlorophyll will be broken down, and plants would then focus on synthesizing accessory pigments that are needed for to give colors to the fruits. And the second one is to produce. Again, it also affects the expression of gene, especially on cellulase and pectinase, which can be used to break down or soften or loosen up the cell wall, which eventually bring about to breaking down of the cell wall itself. And formation of volatile compounds as attractants and conversion of starches and acid to sugar, making the fruit more sweeter. So those are the steps that ethylene supports in order to drive fruit ripening. Okay. So this climacteric response does not occur in all species. However, it is true for apples, tomatoes, and avocados, but not for citrus, grapes, or strawberry. All right. Generally, fleshy fruits can be divided into two groups based on the presence or absence of a respiratory increase at the onset of ripening. So this respiratory increase, which is preceded or accompanied by a rise in ethylene, is called a climacteric. So um, any ripening of the fruits that is accompanied by ethylene, we call it as climacteric fruits. So climacteric is a stage of fruit ripening associated with increased ethylene production and a rise in cellular respiration, and is the final physiological process that marks the end of fruit maturation and the beginning of fruit senescence on. Okay. Non-climacteric fruits ripen without ethylene and respiration bursts. So the ripening process is slower, and for the most part, they will not be able to ripen if the fruit is not attached to the parent plants. So example of climacteric fruits that we have citrus fruits, grapes, strawberry. So example of non-climacteric fruits. Example of climacteric fruits, we have apples, we have bananas, tomatoes. So essentially, a key difference between climacteric and non-climacteric fruits, particularly for commercial production, is that climacteric fruits continue to ripen following the harvest, whereas non-climacteric fruits do not. The accumulation of starch over the early stage of climacteric fruits development may be a key issue as starch can be converted to sugar after harvest. So what does it mean by this? It means that um, in the environment or in our surroundings, there are two types of fruits. We have climacteric fruits, we have non-climacteric fruits. So climacteric is basically the process of fruit ripening uh, when there is, which is induced by increased concentration of ethylene or increased production of ethylene and by increased cellular respiration of the fruit itself. Non-climacteric fruits, it if the fruits is ripening, is not accompanied by increase in ethylene production or increase in cellular respiration. And climacteric fruits can also be induced into fruit ripening even after harvesting. So basically, what it means that when you harvest a fruit which is not mature yet, all right, you pluck it and you put it somewhere, and then you can supply ethylene to it, or you can either envelope it that more ethylene would be produced. And this is what we call as climacteric fruits. Non-climacteric fruits, if we pluck or if we pick the non-mature or non-ripened fruit, when we put it on the bench, we sort of envelope it with newspapers or even paper, it won't ripen. Okay, I hope you guys can differentiate between climacteric and non-climacteric. Right. You can do, back then when I was in Australia, we went for grape picking, somewhere in December, if I'm not mistaken. All right. So we only pluck the ripened grapes because there's no point in plucking non-ripened grapes because it won't ripen when you harvest it. It's not like what does it call, jackfruit. Okay, not like jackfruit where it can, or mangoes, it can still ripen when you harvest it. So that's the difference between climatic fruits and non-climatic fruits. Okay. So ethylene also inhibits flowerings in a lot of species, but promotes only in few species. So it has two functions. It depends on the species of the plants whether ethylene production would inhibit flowering or ethylene production would um, promote or induce flowering. So it also promotes senescence of flowering plants. Abscission, an increase in ethylene production causes the breakdown of the middle lamella, and this results in abscission. So basically, ethylene also support abscission. It's also used to increase the efficiency of harvesting fruits such as cherries and grape. So I think we have also looked into ethylene function in supporting abscission when we talk about auxin. So you guys remember the slides that I have where there's a leaf, and then after that, um, the leaves grows, starts to grow with presence of auxin at higher concentration, and this would decrease the sensitivity to ethylene. And then when the leaf starts to wilt, so we can see that there's an increase in the sensitivity of ethylene. So production of ethylene would induce abscission. And ethylene is also used for abscission in terms of increased efficiency of harvesting fruits. So if the fruits has already been ripened, or if the fruit is uh, ripened fruits, all right, so we can apply ethylene in order to induce the harvesting, in order to induce the abscission of the ripened fruits itself. So sex expression, the sex of the flowers on monoecious plants contain both male and female flowers is determined by the concentration of gibberellic acid and ethylene. If more gibberellic acid is being produced, it's known as male flower. It would incline more towards becoming a male flower. And why is it important to determine male flower and female flower? It's basically for fertilization of the plants. And then ethylene production at high concentration, it would cause the formation of female flower. So it is used to synchronize flowering in pineapple to get that perfect shape. So we can manipulate ethylene in order for us to increase the production of gibberellic acid, to have more male flower, or increase the production of ethylene to have female flower. So we can synchronize the flowering in pineapple to get that juicy, perfect shape of pineapples. And stem elongation, shaking increases ethylene production, which causes cells to grow along with curved stems. So ethylene has a lot of functions. And again, just like gibberellic acid, it also works together with auxin. And these are the processes that I think it's one of the important roles of ethylene, which is supporting food ripening. And what are the processes that occur in order to support that food ripening itself? Now we move on to our last important hormone, which is called abscisic acid. Abscisic acid, the name itself, because of the name, is often referred to as an inhibitory hormone rather than stimulatory hormones such as ethylene, uh, auxin, gibberellic acid, and also cytokinin. It is found in leaf where it is partially synthesized, stems, and can also found in stem and green fruits. And its biosynthesis is related to the process of carotenoid production. It is generally associated with negative feedback interaction or stress-related environmental sickness such as drought, freezing temperature, and environmental pollution. So basically, abscisic acid is being produced when plants are challenged with stress. And stress for plants are basically because of the temperature. Uh, so drought, freezing temperature, environmental pollutants, drought and freezing temperature, basically at extreme condition, abscisic acid is being produced. It's part of the defense mechanism of plants. So what did abscisic acid do in order to as a defense mechanism in extreme condition? And this is how it looks like, the abscisic acid. Okay, so we can see the carboxylic acid group. That's why it is uh, in acidic form. All right, in acid form. So closure of stomata in extreme condition, in high temperature, um, high heat, or intense sunlight. So large amount of acetic acid in the leaf causes the stomata to close, which help the plants to conserve water during droughts. The reaction can be instigated within minutes of spraying. Commercially, it is used in field when drought threatens. So basically, so we have different types of plants. So you guys have learned about different. I'm sure, I believe you guys have learned on different types of plants, maybe in plant biochemistry, or you also learn it when you learn about photosynthesis, how plants adapt to the environmental stress, drought, or extreme condition, intense sunlight, uh, too high of a temperature. So what's going to happen? So one of the things when plants are exposed to drought or extreme condition of high sunlight and high temperature, so it would induce the closure of stomata, right? When the stomata is closed, basically it avoids or it prevents the loss of moisture. So plants will keep the moisture when the stomata is closed. So that's why we have different types of plants. We have C3 plants, we have C4 plants, and we have CAM plants. And it all depends on the temperature. So extreme temperature in the desert, we have the stomata being closed uh, during the day, and it will only happen. It will only opens up during the night, right? So abscisic acid induce closure of stomata itself in order to uh, preserve the moisture in the plants during extreme conditions such as drought. And bud dormancy, this is influenced by cytokinin and auxin-induced synthesis of ethylene. So it basically causes or prolong bud dormancy. With because we know that abscisic acid is an inhibitory hormone. So it's like a defense mechanism for um, plants during extreme condition, right? And it induces or it prolongs bud dormancy, but will only germinate when it's the right time to germinate, right? When the environment or the surrounding is less dangerous to the plant itself or to the seed. So this is what I told you guys. Acetic acid delays seed germination in many plants, has some effects on induction and maintenance of dormancy. In general, it induces seeds to synthesize more storage protein, more starch, in order to supply food. Okay, so instead of breaking down the storage protein or storage carbohydrates, it basically synthesize more of this in the endosperm. So it inhibits the action of gibberellin. So gibberellin, just now we know that the effect of it is to break seed dormancy. So when abscisic acid, when it sends the extreme condition, it will tell the plants or it will tell the embryo not produce any more gibberellic acid. So we have to keep on being dormant. So produce more and more abscisic acid. So its impact decreases when seeds germinate. In desert plant, ABA found in the outer layer of the seeds must be washed away by rain in order for the seeds to germinate. So this is how it looks like. Apparently, it can be dissolved in water. So it counteracts stimulatory effects of other hormones. One example that we have is it inhibits the production of gibberellic acid, and it inhibits shoot growth. It induces gene transcription. It inhibits shoot growth, but will not have as much effects on roots. May even promote growth of roots. It inhibits cell division in the vascular cambium. So basically, it promotes growth of roots because when the root is growing, it basically can absorb more nutrients. Uh, so it induces gene transcription, especially for proteinase inhibitor in response to wounding. So if there is any response, if there is any wounding in the in the plant itself, it will induce proteinase inhibitors. It's basically one of the defense mechanism in plants to avoid any invasion of pathogen. So this one here is what vascular cambium is all about. So it's the main meristem in this. It's the main meristem in the stem, producing undifferentiated root cells inward and bark cells outwards. Right. Let's just go for a short 5 minutes break before I continue with other substances that can also be considered as hormone. And if you have any question, you can either ask them now or you can type it in the chat box and then I will answer it later after we finish our break. All right. So we're going to continue with the rest of the topic on other additional substances gaining recognition as a hormone. So this is in addition to the five hormones that we have talked. All right. So we talk about auxin, we talk about gibberellic acid, talk about cytokinin, ethylene, and we sort of end it with abscisic acid. So now we're going to talk about other additional compounds which also gain uh, attention, all right, as hormones. So one of them is steroid. From the name of it, we know that it's a steroid and hormone. It's made up of steroid. So there are approximately 16 steroidal compounds known as steroid, named after the first one identified, which is known as prasinolite, which was found in mustard pollen. So what does prasinosteroid do? It stimulates stem elongation, it inhibits root growth and development, and promotes ethylene biosynthesis and epinesty. So the most important or the most distinct function of prasinosteroid is what we call as epinesty. We don't found it in auxin, we don't found it in gibberellic acid, ethylene, or cytokinin, right? But if you still remember on the first few sites, all right, we talk about how um, multiple hormones are working together just to support one response of the plant on one reaction in the plant. So it's the same thing here, right? So we know before this we have encountered hormones that also support stem elongation, also support, also inhibit root growth and development. So this one here, leaf epinesty involve the downward bending of leaf as a result of disturbance in their growth with a greater expansion in adaxial cells as compared to abaxial surface cells. So this is how it looks like. So weeping willow is a state of permanent epinesty. So it produces a lot of prasinosteroid. So instead of growing upwards, it grows like downwards. It form an epinesty kind of thing, which is downward bending of leaf as a result of disturbance in their growth because they have prasinosteroid production. And we have salicylate, comes from the word salicylic acid. Salicylates have been known to present in willow bark. Salicylic acid is synthesized from the amino acid phenylalanine. If you um, are familiar with it, if you always buy your toiletries or your wash products, you can have a look at the ingredients, and one of them that we always find is salicylic acid. So effects include thermogenesis in arum flowers, plant pathogen resistant. It stimulates plant pathogenesis protein production, and it is reported to enhance longevity of the flowers. So you don't really have a lot to know about this one. Um, this is just the least of what salicylic acid do. All right, and it is reported to inhibit ethylene biosynthesis, reported to inhibit seed germination, blocks the wound response, reverses the effects of ABA. We don't really talk in detail about this because it's very, very complicated because it involves a lot of reactions at one time and a lot of involvement of other hormones also for salicylate. But it does have a wide variety of um, functions. And then we have jasmonate. Uh, it first isolated from the jasmine plant, jasmonic acid, right? In which the methyl ester is an important. in which the methyl ester is an important product in the perfume industry. So anything that is ester, it gives you a good smell, a good fragrance, right? And jasmonic acid is synthesized from linolenic acid, which is an unsaturated fatty acid, which is an important fatty acid. Jasmonate have a number of effects, such as, so basically jasmonates are the one that produce jasmonic acid, and jasmonic acids are basically used as a form of fragrance, and it inhibit many processes such as growth and germination. It promotes senescence, abscission, tuber formation, fruit ripening, pigment fermentation, and then recoiling. So they appear to have important roles in plant defense by inducing proteinase synthesis, just like abscisic acid, defense mechanism against fungi. So this is jasmine flower, and from jasmine flower, that's where the jasmonic acid is being first isolated. Right. So that's about it for our plant hormone. So I don't really stress a lot on other hormones, despite that five hormones that are important.
Recording stopped.
Right. So I'm just going to recap back what we have learned, and then later on I will open Q&A session. If any of you has any question on plant hormone itself. Right. Let me just open my OneNote first. So this is one of the things that we learned just now. This is the role of gibberellic acid. So you should be able to explain the role of gibberellic acid in seed germination, in breaking up seed dormancy. So gibberellic acid, uh, so this is in aleuron cell or aleuron layer, aleuron cells in the aleuron layer. So GA being produced will then go to the endosperm. Starch. So this is what happened in a seed normally, without any, without any uh, exogenous GA. This is what happened. A normal GA release from the embryo itself, and then it would induce starch hydrolysis, producing maltose, and this maltose will then be used for energy generation for the seed to break from dormancy, right? And industry manipulate these, right, by supplying exogenous GA to increase the rate of uh, seed germination, right? And a rapid production of carbohydrates or monosaccharide or disaccharide, all right? Maltose. And it is then manipulated in the beer industry, right? Using barley seed, where this maltose here is then channeled to full of yeast, all right? It can either be in bioreactor kind of form or just in normal bucket of yeast, and then yeast would undergo alcoholic fermentation to produce ethanol, right? So you have to remember what is alcoholic fermentation, what is the enzyme involved, and what actually happens. It's basically pyruvate being converted into acetaldehyde and then being converted into ethanol. And this is what they manipulate in terms of the beer industry. And gibberellic acid can cause the increase in expression of gene for different types of amylases. So we have identified how many four amylases with different PI value, right? So when it has different PI value, it will then be activated at different times, all right? But it basically uh, ensure there is a continuous supply of amylases so that more and more maltose can be channeled to the yeast for alcohol fermentation. So this is what we have talked for this about gibberellic acid. And you guys also have to know about the difference between um, gibberellic acid and auxin, right? And you know that gibberellic and auxin, it works together. They work together and athenocopy, right? Again, just check the um, the spelling, whether it's a correct spelling or not, right? So production of fruits without fertilization or pollination. This one here supports growth, right? So the flower can be big, the flower can be bigger, the plants can be bigger, and also the fruits can be bigger with the application of exogenous gibberellic acid. So that's what gibberellic acid does. And then we have looked into another hormone, uh, a cytokinin hormone. Why is it called cytokinin? So it supports cell division, right? And you guys can refer to the section note how it supports cell division, right? Where is it? So it supports cell division. It basically increase the rate of conversion or transfer of G2 phase into mitotic phase, right? And um, that's the role of cytokinins, and it also has a role in formation of organs and delaying senescence. And then we have talked about another hormone is known as ethylene. In ethylene, it's very familiar with fruit ripening. So what are the processes involved in driving the ripening of the fruits? All right, breakdown of chlorophyll and production of accessory pigment to support the color production in fruits. So, and then production of more cellulase, like kinase, that breaks down the cell wall and conversion of starch into called as sugar. Conversion of starch so that the ripened fruit will be more juicier, more sweeter. Starch too, right? A formation of volatile compound that give a good smell uh, to the expectant for the process of pollination or to the human nose itself, like a pleasant smell, right? So this volatile compound is basically like a lipid-based compound, right? Which are volatile. And then you also have to know what are the experiment, what was the first experiment that leads to the identification of ethylene itself. And what is climacteric and non-climacteric foods. Okay, what's the difference between this, and is there any different in terms of the ethylene production and things like that? And it also promotes abscission. Okay? And it works together with auxin. So lower concentration of auxin will increase the sensitivity towards ethylene, and because of that, it is also manipulated in terms of fruit harvesting in agriculture, right? Okay, and we have also looked into other functions, but I would say these are the important functions of ethylene. And then we look into abscisic acid. Why is it named abscisic acid? Because it has um, what we call as inhibitory property, right? Abscisic acid. Abscisic, ABE, right? It's known as a plant defense hormone. In extreme weather condition, in freezing, extreme weather condition, drought, freezing temperature, or even environmental pollutants. So what does it do? It does the stomata, basically prolong bud dormancy. So the seed won't produce gibberellic acid that basically is involved in uh, breaking dormancy. And we have also looked into other substances or other compounds that can be considered as enzyme also. So brasinosteroid, mostly in creating epinacity. And then we have jasmonic acid, um, basically, or jasmonate, right? Jasmonate producing jasmonic acid, which is also known as a hormone that produce fragrance. And then we have also looked into salicylic acid. That's it. So that's all about uh, plant hormone. Any question? Degrees of the test two. Apparently, Dr. Izwan has your answer scheme. I'm still waiting for him to give it to me so that I can mark my parts. Hopefully, by before you guys go into your final exam, you can know the marks for your test two, and Dr. Izwan also should be able to key in the marks for your SCL in the ESMP by this weekend, right? I will try and talk to him. I will try and ask him whether he can give it to me ASAP for the test two answer marks or answer sheet. Okay, Malvina. All right. Any other question? If there is no question, I guess your test would be on the 30th, isn't it? 30th January, which is next Monday. Um, the same format. Yeah. Um, maybe two or three essay questions that you guys have to answer. It's the same format, the same overall marks. For your test two, the overall marks was basically 80%. All right. And from there, we take only 20% for your carry mark. It's the same thing with your final. It's just that your final, I think it makes up about 30% of your overall marks, if I'm not mistaken, because your test one, 20%, test two, 20%, 40. And then you have done like two SCL with me. The first one is your poster on diabetic. That one will also be graded. And the SCL that you guys done with Dr. Izwan for your mini exhibition, that would account for 20% of the overall marks. And 20 is 60. So 40% for your final. I think it's written in the ESMP, if I'm not mistaken. Right. Um, I hope that answer your question, Adila. Any other questions? If there are no more questions, I guess that's our final. This is our final lecture. So I tried to key in your attendance last night, but I couldn't open the ESMP. But I have counted your attendance manually, and there are about 3 person that wouldn't have 80% of attendance. You can check it by today, by tonight, or your attendance, whether you achieve 80% or not. And I would also give the names to Sopita so that Sopita can announce it or message you personally, right? Who has the attendance that is less than 80%. So again, if you have less than 80% and you don't have any concrete reason or concrete justification for it, no MC or medical leave certificate, so you won't, I won't be able to be, basically I won't uh, treat it as um, a reasonable excuse if you don't have um, medical certificate for that. If, say, for example, you are absent because you are not well. Okay, right. If there is no question, I guess that's it for me from me. Another thing about next semester subject, um, the animal tissue biochemistry. I know that in ESMP, it is stated there that I'm one of the lecturers that is going to teach you, but there has been a change in that. Dr.